Hi everyone. Today let us discuss which are the most favorite microbiological tests used to test the water potability or the property whether it's fit for drinking or not. This method has three parts: presumptive test, confirmed test and completed tests. The first part is presumptive test or most probable number method, MPN, which presumes that there is fecal contamination in the water sample, meaning that the water is not fit for drinking and it should be further treated. It is a very simple, easy, reliable and inexpensive method. Here the water sample is aseptically inoculated in quantities 0.1 ml, 1 ml and 10 ml in 3 tubes each of 10 ml lactose fermentation broth and are incubated at 37 degrees for 24 to 48 hours. This lactose fermentation broth includes lactose, beef extract, peptone and phenol red indicator, which is orange in the original broth at neutral pH but turns yellow in acidic conditions. So if there is fermentation of lactose to lactic acid, it means if there are lactose fermenting bacteria present in the water, then we will see it as a color change. Further, because these produce gas also, so there will be gas produced in the system and it will be visible to us as a bubble in the Durham tubes, the small tubes which are inside these test tubes. Before autoclaving, these small tubes, Durham tubes, are inverted into these tubes, inserted and inverted, which are useful in trapping any gas that's produced by the microbes present in the water sample. The 0.1 and the 1 ml water sample is inoculated in the single strength broth, and the 10 ml water sample is inoculated in 10 ml of double strength broth in a thick test tube called the boiling tube, thereby converting the double strength broth into single strength automatically because we add 10 ml of the water sample to 10 ml of double strength broth. Now, if the water being tested has fecal coliforms such as E. coli, these microbes will ferment lactose and make lactic acid that will result in turning the broth yellow and production of gas in the Durham's tubes. Since this method is statistical, it has to be conducted in replicates. Ideally, it's 3 or 5 copies of each tube, making it a total to be 9 tubes or 15 tubes respectively. After incubation, these tubes are observed for acid and gas production. This method is based on probability statistics, hence replicates of the tubes are needed. MPN per 100 ml is not a direct count, rather it's the most probable number based on the statistics. Now, when we observe these tubes and find the positive result in any tubes, positive here means acid plus gas, both together, then it is counted as positive test, and this is matched with the ready reckoner, a table which indicates how many cells are most probably present in the given water sample. In India, 2 to 3 living cells per 100 ml of water and 0 living cells per 100 ml in developed countries is the norm, and from this matching the results, one can identify whether this water is potable or not, can it be safely consumed by people or not. Indicators of water safety should be easily and quickly detectable, easily distinguishable from other food biota, should have association with pathogens, should always be present in detectable numbers. Their numbers should ideally correlate with those of the pathogens of concern, should have better growth rate than the pathogen, should have a die-out rate at least equal to that of the pathogen. Ideally, they should persist for more time, should be absent from the foods which are free of the pathogens. So all these criteria are qualified by Escherichia coli. The history behind this Escherichia coli as the indicator microbe goes like this. In 1885, Escherich, while attempting to isolate the etiologic agent of cholera, isolated and studied the bacterium that suited the bill to be the indicator microbe. It was named E. coli after the discoverer's name, and coli because its origin is there in human intestines or large intestine, and so this was taken to be the ideal indicator microbe that was different from Enterobacter aerogenes, which is found on plant surfaces and in soils. Now, once we determine the MPN and we get any test tube which shows a positive result of acid and gas production, from there we go on to do the confirm test. A loopful of water is taken from the positive MPN tube and streaked onto EMB agar, eosin methylene blue. If it grows as a colony, which is with a green metallic sheen, it shows the presence of bacteria directly. So this result in finding out that indeed Escherichia coli was present in the test tube, showing the positive result of acid and gas production. So this is the confirmed test because we now confirm the presence of E. coli in the positive test tube. And once again, to be 100% sure, a third and last test, completed test, is conducted where a loopful from the isolated colony with the green sheen is taken and aseptically inoculated into the fresh similar lactose fermentation broth. If the same result is reappearing, that is, the reproducibility of the results is seen, if the broth turns yellow and a gas bubble appears in the Durham's tubes, it is a positive completed test. Also, from this, a slide is made by the gram staining, and the presence of gram-negative non-spore forming rods from the agar gland constitute a positive completed test. My dear audience, these tests are very simple and reliable, though time-taking. In subsequent videos, we shall bring for you more methods for water potability. Meanwhile, if you have any questions, feel free to ask or comment about what you felt about this video and your feedback. Take care. It's bye till next time.